So one question that comes up with these myeloma progenitor cells is whether they contain all the point mutations or single nucleotide variations that are present in myeloma plasma cells. And we attempted to answer this by doing a combination of whole exome sequencing and patient-specific custom capture targeted deep sequencing of the B-cell subpopulations to a depth of about 20,000x. And by doing this, we're able to see that many of the single nucleotide variations that are present in the myeloma plasma cells could also be seen in the pre-plasma cells and B cells, although at a much lower allele frequency. So then the really critical question is, do these cells contribute to relapse in myeloma? And to answer this, we tried to follow these subpopulations of cells in patients over time. And we did this by taking serial bone marrow samples from patients before, on, and after treatment, and then sorting out these different B-cell subpopulations from the bone marrows at each of these time points, and then identifying the mutational signature of each of those subpopulations to answer the question, do the plasma cells pre-treatment give rise to the plasma cells after treatment, or do the plasma cells after treatment arise from some other population? And we reviewed 3 different patient results with multiple bone marrows and subpopulations that had been sequenced. In all 3 cases, we found that the plasma cells at relapse were missing some of the mutations that were present in the plasma cells pre-treatment and had instead developed new mutations that weren't present in the plasma cells pre-treatment, but which could be detected in the myeloma B cells prior to treatment, often, even up to 3 years earlier. So our conclusion from that work was that in many cases, the myeloma plasma cells at relapse can arise from the myeloma B cell compartment pre-treatment and not necessarily from the myeloma plasma cells. And those myeloma B cells can be CD38 negative, CD138 negative, and also can be BCMA negative. So this has important implications for myeloma treatment. We think to cure myeloma, we really need to, if we want to eradicate the entire clone, we need to be able to target these myeloma B cells. So in conclusion, myeloma is a disease that probably in many cases originates in the germinal center, where many of the clonal driver genomic events arise and where even the clonal single nucleotide variations arise. The germinal center outputs both plasma cells and B cells. And in myeloma, we treat the plasma cells, but we don't treat the B cells. And the B cells are left behind. They're a non-proliferative population. They're non-secretory. But when they mature into plasma cells, their oncogenes can start to be expressed, and they become proliferative, and they can recapitulate the disease. So we think in myeloma that we have to go after those earlier populations of cells in addition to myeloma plasma cells, and hopefully one day that will lead to a cure.